The biosphere is packed with an overwhelming density of living material. From microscopic bacteria in the soil, to apex predators, to the massive root systems holding the forest floor together, biology at first glance looks like pure chaos. To make sense of this, we use taxonomy. It is an analytical framework that allows us to map out exactly how biological capabilities evolved over time. But to build a system that classifies all living things, we need an absolute baseline. We need a strict, objective boundary that separates one type of organism from another. We need to define a species. This diagram illustrates the biological test for a species. Two organisms belong to the same species only if they can successfully mate and produce an offspring that is also capable of reproducing. A lion and a tiger can physically mate to produce a liger. However, because that liger is genetically infertile, the biological line stops there. This reproductive dead end confirms that lions and tigers are entirely separate, distinct species. This proves that taxonomy is built on objective, structural functions like reproduction and genetics, rather than subjective visual similarities. Historically, early thinkers completely missed this level of functional detail. Aristotle, for instance, organized plants into 3 simple bins based entirely on their height: herbs, shrubs, and trees. That system grouped entirely unrelated organisms together simply because they happened to grow to the same size. It was a model based on the naked eye, and it fell apart under scientific scrutiny. This is R.H. Whitaker's 5-kingdom classification grid. Instead of looking at the outside of an organism, Whitaker's model evaluates the internal architecture of its cells. By tracking variables like whether a cell has a distinct nucleus or whether the organism operates as a single cell or a complex multicellular system, the model systematically filters out the simpler life forms. We can isolate the kingdom Plantae from remaining groups, like fungi, by identifying one highly specific piece of biological hardware, the chloroplast, which allows plants to manufacture their own food through photosynthesis. Now that we have isolated the true plant kingdom, we can track how plants conquered the planet by mapping their evolutionary hierarchy from the ground up. This checklist displays the primary structural upgrades. It maps 2 major divisions: cryptogams using hidden spores, and phanerogams producing visible seeds. At the absolute baseline, we have Thallophyta, or algae. Notice every single box remains completely empty. Thallophytes live almost entirely underwater. They have no true roots, stems, or leaves. Their entire body is simply an undifferentiated mass of tissue called a thallus. As we step onto land, we reach the division Bryophyta, which includes mosses. The main structural checklist is still blank, but they develop primitive, thread-like anchors called rhizoids. Biologists refer to bryophytes as the amphibians of the plant kingdom. While they can survive in moist soil on land, they still require physical standing water to carry their reproductive cells. Because they lack true roots and have no internal tissue designed to channel moisture, their anatomy restricts them to incredibly small sizes. Without a biological plumbing system to pull water upward against gravity, these early cryptogams are physically incapable of growing tall. The arrival of the division Pteridophyta, the ferns, introduces a major structural upgrade. For the first time, plants develop true roots and vascular tissue. This internal plumbing network acts as an intricate circulation system, finally allowing plants to transport resources over long distances vertically. The system relies on 2 distinct transport lines: xylem, which pulls water up from the roots, and phloem, which distributes the food created in the leaves down through the rest of the plant. Yet, despite having true stems, roots, and leaves, ferns are still classified strictly as cryptogams. They reproduce through spores, not seeds. A spore is incredibly simple. It is a single cell ejected into the wind, carrying 0 stored food, leaving its survival entirely to chance. Vascular tissue gave ferns the physical strength to conquer dry land, but their microscopic, vulnerable spores kept their reproductive success rates incredibly low. To solve that, evolution crossed a major taxonomic divide into the phanerogams. This is the arrival of the seed, seen first in the gymnosperms. Instead of relying on a fragile spore, a gymnosperm packages a developed plant embryo inside a protective casing, along with a dedicated food supply. This completely removes the plant's reliance on standing water for reproduction. This photograph of a giant sequoia demonstrates the sheer physical scale made possible by combining thick vascular tissues with the reproductive autonomy of seeds. However, gymnosperms like pines and cycads produce what are called naked seeds. They develop exposed on the surface of cones, without any outer fleshy layer. The final structural upgrade occurs in the division angiosperms, where the last box on the biological checklist is successfully filled. Angiosperms engineer flowers to hijack insect behavior for highly targeted pollination. They then encapsulate their seeds safely within the dense protective tissue of a fruit. That enclosed, heavily armored seed represents the absolute highest tier of botanical engineering. Even within these highly advanced angiosperms, structural efficiency drives one final taxonomic split. We categorize them based on the anatomy of their seed leaves, known as cotyledons. The first category is the monocots. Their seeds contain exactly one cotyledon. That single internal difference dictates the plant's entire external architecture. Monocots develop shallow, fibrous root systems, possess hollow or false stems, and grow leaves marked by perfectly straight parallel veins. This archival photo of a grain harvest shows how this hollow-stemmed monocot structure is heavily leveraged in global agricultural staples like wheat, maize, and bamboo. The second category comprises the dicots, whose seeds are packed with two distinct cotyledons. Look at this photograph of a cotton field. The dicot seed produces a totally different biological structure. They develop a deep central taproot, dense solid stems, and broad leaves driven by complex net-like veins. In this photograph of a banyan grove, we see the ultimate expression of the dicot blueprint. The combination of solid wood and deep taproots allows them to build sprawling, incredibly dense architectures that can stand for centuries. Knowing whether a seed is a monocot or a dicot instantly reveals the plant's entire mechanical strategy, right down to the density of its cellular walls. We can read plant taxonomy as a direct map of evolutionary adaptations. We track the progression from an aquatic stylus over amphibious mosses, through the vascular plumbing of ferns, then through efficient gymnosperm seeds, finally peaking in the flowering architecture of angiosperms. Botanical classification serves as a decoded record of how life iteratively solved the physical challenges of